Hello friends, today let us discuss the chromosome structure, classification and ultrastructure of chromosomes. I am Zaiba Nishad, Head of the Department of Botany, Vijay College, Bangalore. Now by chromosome structure, we mean the structure of the eukaryotic chromosome. Now let us see how actually the eukaryotic chromosome appears. You can see here in the first diagram, you can see a complete chromosome. You can see that there is a white dot in the center. Now that white dot in the center is nothing but the centromere, which is otherwise known as the primary constriction. Now this centromere holds together two chromatids. You can see two chromatids there. One chromatid as being shown by the arrow from the top to the bottom, that is one chromatid, and on the other side you can see the other chromatid. So there are two chromatids here. Now coming to the next diagram, chromatin thread. Now by chromatin thread we mean thread-like structures, chromosomes which are thread-like structures. Generally in the normal cell you find thin thread-like structures. These thread-like structures are called as chromatin threads. For example, in a human cell, the diploid number is 46. That is 2n is equal to 46. So you have 46 thread-like structures, or in other words 23 pairs of thread-like structures. So you have a human diploid number 2n is equal to 46 or 23 pairs of chromosomes. Now, if you take the expanded chromosome as in the next diagram, that is the diagram that is got after DNA duplication. When does DNA duplication happen inside the cell? Please remember we have already studied mitosis and meiosis. In both the types of cell division, you have an interphase. The interphase which is divided into the G1 phase, S phase and G2 phase. The S phase is known as the synthetic phase. Now in this synthetic phase, there is the duplication of the DNA. So that single chromatin thread after DNA duplication forms two threads. See in this expanded chromosome, you can see two threads held together by that orange dot. That orange dot is that centromere. Okay. So you can see after DNA duplication, you have these two thread-like structures which on condensation, as in the third diagram, that condensed chromosome, these two threads they condense highly. Once they are highly condensed, you get these chromatids. Chromatid is nothing but a condensed chromatin thread. Am I clear now? Condensed chromosome is nothing but it has two chromatids as you can see here. Now these two chromatids have a very highly condensed chromatin thread-like structures. Now in the last diagram you can see some beads, nucleosomes in the chromatin. So these beads are nothing but the nucleosomes. These nucleosomes are also known as the new bodies. We will come later how actually a nucleosome is organized. Now what are these chromosomes? I already told you it has a coiled thread-like structure. That coiled thread-like structure is also called as the chromonema. Why it is called chromonema? Because when you stain it, it takes up color. So it is called as the chromonema. Now each chromonema has major and minor spirals that we will understand when we do the nucleosome model of the DNA. Next, now these chromonema contains genes, and you all know, you have already studied genetics. These genes are arranged in a linear order on the chromosome, and you can study the chromosome best during the metaphase and the anaphase stage because the chromosomes are highly condensed. The chromatin material is highly condensed, so you can study the chromosomes very clearly in these two stages. In the metaphase chromosome, as we saw in the previous slide in the first diagram, you can clearly see two chromatids held together at the centromere region, and each of this chromatid has the chromoneme, which is nothing but the stained portion of the chromosome. The chromonemata are involved in the transmission of the hereditary characters. Chromonemata, I repeat again, expanded form is the chromatin thread, condensed form is the chromatid. Now, anywhere along the length of the chromosome, you find a weakly staining or a clear constriction. By constriction, you mean a narrowing, which is called as the centromere or the primary constriction. In a metaphase chromosome, the centromere is made up of two C-shaped structures. Each centromere is made up of two C-shaped structures. These C-shaped structures are called as kinetochores. What's the function of these kinetochores? They get attached to the chromosomal fibers that help in the movement of the chromosomes towards the poles during cell division. Now, in some chromosomes, not in all the chromosomes of the cell, you also find one more constriction. That constriction is called as the secondary constriction or is called as the secondary constriction. Now, let us understand this. So, apart from the primary constriction, you have one or sometimes more than one constrictions which are called as the secondary constriction. Now, the secondary constriction is called as the nucleolar organizer. We have studied that in cytology. Why it is called as the nucleolar organizer? Because it helps in the formation of the nucleolus. You know, the nucleolus is a small, dense portion inside the nucleus of a eukaryotic cell. Now, the region of the chromosome beyond the secondary constriction, it is called as the satellite, abbreviated as SAT, S-A-T. Satellite, it is a spherical body connected to the rest of the chromosome by a slender stalk. Satellite is also known as trabant. Now, I'll repeat again. There is the primary constriction, as you saw in the previous slide, which is holding the two chromatids, that is the primary constriction. Now, sometimes in some chromosomes of a cell, not in all, you find one or more constrictions which are called secondary constrictions. Now, the portion of the chromosome beyond the secondary constriction is called as the satellite. Now, the ends of the chromosomes are generally degraded by nucleases which cause aging or senescence. And to prevent this, eukaryotic chromosomes contain repetitive non-coding sequences of the DNA with proteins. So, this repetitive non-coding sequences of the DNA present in eukaryotic chromosomes are called telomeres. Now, what do the telomeres have? They have thymine and guanine paired with adenine and cytosine. In germline cells and in cancer cells, there are telomerases that replace the lost ends of the DNA and prevent aging. So, in germline cells, what do you mean by germline cells? In the reproductive cells. And cancer cells, only because these are replaced, perhaps they tend to become immortal. Now, the chromosome bearing the satellite is called as the SAT chromosome. What does SAT stand for? It stands for sin acido thymonucleonico. That means it is a portion of the chromosome where there is no thymonucleic acid in the DNA because of relative deficiency of DNA in the nucleolar organizer region. The significance of this satellite is not yet understood. The terminal regions or the ends of the chromosomes are non-sticky and they are called as telomeres. I already told you what are telomeres. Now these telomeres are the ends of the chromosomes. They contain tightly coiled and densely staining chromatin. This tightly coiled and densely staining chromatin, we call it as heterochromatin. Telomeres maintain the integrity and individuality of the chromosomes because they prevent one chromosome from sticking with the other chromosome. Now coming to the classification of the chromosomes. Here to my right, you can see 4 diagrams. The shape of the chromosome is varying according to the position of the primary constriction. So based on the position of the primary constriction of the centromere, we have 4 categories of chromosomes. The first one is called as the telocentric chromosome. Now telocentric chromosome is the last one, D, the figure D there in the diagram. What do you mean by a telocentric chromosome? Here the centromere is placed at the end of the chromosome. It's placed at the end of the chromosome. Hence this chromosome appears eye-shaped during anaphase. So it appears to have only one arm. So hence it is called isobrachial. The second category is acrocentric chromosome, which is represented by the diagram C. What is an acrocentric chromosome? Here the centromere is placed slightly away from one of the ends of the chromosome, or in other words, the centromere is subterminal in position. It's not at the terminal end, not at the end, it is subterminal, slightly away from the end of the chromosome. So how does this chromosome appear when it is pulled during the anaphase stage? It appears J-shaped. This type of acrocentric chromosome is also called as cephalobrachial chromosome. The third category is the submetacentric chromosome, which is shown by the figure B. A submetacentric chromosome is called as a heterobrachial chromosome. Heterobrachial, two arms which are of different sizes in length. That's the meaning of heterobrachial. Now where is the centromere placed here? The centromere of the primary constriction is placed slightly away from the center of the chromosome. That's why it's called a submetacentric chromosome. It is placed away from the center of the chromosome such that there are two unequal sized arms, a long arm and a short arm. You can see here in the figure B, there's one long arm and there's one short arm. So this forms the submetacentric chromosome, and when it is pulled, you can make out there, I have already shown it in the bent form, it appears as if it is shaped as the letter L during anaphase. Pulled by what? Pulled by the mitotic spindle or the chromosomal fibers. Next, the metacentric chromosome. It is called as isobrachial because it has two equal arms. And where is the position of the centromere here? It is exactly median in position, or it is placed at the center of the chromosome. Hence, the chromosome appears V-shaped during anaphase, as you can see in the diagram. Now coming to the classification of the chromosome based on the number of centromeres, you have acentric chromosome where the chromosome is complete, where the centromere is completely absent. You have a monocentric chromosome as we just now studied, which are the most common types having a single centromere. Dicentric chromosomes which has two centromeres. Polycentric chromosomes are chromosomes which are having more than two centromeres. They are very common in birds and moths. Then diffuse centromeric chromosomes. In this type, the centromere is not organized into a distinct structure. It spreads over a certain length of the chromosome, so appearing diffused. This type of chromosomes are called as the diffuse centromeric chromosomes. Now coming to the ultrastructure of the chromosome, I already told you we are studying the ultrastructure of the eukaryotic chromosomes. Now the entire eukaryotic chromosome, what is it composed of? About 40% DNA, deoxyribonucleic acid, and 60% of the chromosomes are proteins along with traces of RNA. Now what are these proteins? These proteins are called as the histone proteins. And all these histone proteins, they are positively charged, and we know from our earlier knowledge that DNA is negatively charged. Now these histone proteins are associated with the DNA. These histone proteins are basic proteins and rich in arginine and lysine. Arginine and lysine are basic amino acids. How many classes of histones we have? 5 classes. What are those 5 classes? H1, H2A, H2B, H3 and H4. And which is the largest of these 5 classes of histone proteins? H1 is the largest. Now who proposed the ultrastructural model of the chromosome? It was proposed by Kornberg, Roger Kornberg. He proposed the ultrastructural model of the chromosome. And this ultrastructural model, as I had shown you in the first slide, it shows or represents or explains the chromosome as a string of beads. Now let us understand what actually this string of beads is. Now you can see here, each bead is a nucleosome. You can see in the first diagram. In the very first diagram, you can see some spherical structures, 4 on the top, 4 below. So totally 4 plus 4, 8. They are the 8 histone proteins. which are your uh, which 8 histone proteins which totally form an octamer, 2 H2A proteins, 2 H2B proteins, 2 H3 proteins, 2 H4 proteins. So all these form 8 units of one component, that is the nucleosome. Along over these 8 units of the histones which are compactly held, you have two rounds of the DNA. You can see the red colored DNA forming two spirals. The DNA runs over it in two spirals and comes out. And the DNA that comes out is called as the linker DNA. You can see in the second diagram, the DNA is coming out and that DNA is called as the linker DNA. So it links one histone octamer with the next adjacent histone octamer, and the two rounds of the DNA which surround the histone octamer are tightly held with the help of histone 1 protein. I told you the largest of the histone proteins is H1. So this H1 histone protein holds or binds the two strands of the two rounds of the DNA that move around the histone octamer. Now after this two rounds of the DNA are bound by the H1 histone protein, the DNA that leaves this octamer is called as the linker DNA. Now this linker DNA now goes and links another octamer. You can see in the figure on the top, you can see the next octamer is there in the middle, which is shown as a core of 8 histone molecules. The yellow structure, the yellow one which is binding the two rounds of the DNA, is the histone H1 protein. So similarly there in the diagram you can see 3 octamers which are joined one to another by the DNA encircling it. Each histone octamer is encircled by the DNA twice. Then the DNA strand comes out, encircles, goes 2 rounds around another histone octamer, comes out as linker DNA, again goes around, and these 2 rounds are held tightly onto the histone octamer by the histone H1 protein. So like this continuously, this is the way the proteins and the DNA are organized to form the nucleosomes, and it is these nucleosomes which appear like beads. Your string is nothing but the DNA. So the entire chromosome, the eukaryotic chromosome, has this model. That is the nucleosome model of the DNA, which was proposed by Kornberg. Now in 1975, Autet and his colleagues suggested the name nucleosome to this bead-like structure. So these nucleosomes are the repeating units found in the chromosome. And where can you see this? Of course, under the electron microscope. Now let me summarize once again. Each nucleosome contains a histone polymer. Now this histone polymer is also called as a new body made up of 2 molecules each of H2A protein, 2 H2B proteins, 2 H3 and 2 H4 histone proteins. So totally how many units you got? 8. Hence a nucleosome is an octamer. And the nucleosome also has the double helix DNA molecule coiled around the histone octamer, forming 2 coils, 2 rounds around it. 1/3/4, 2 rounds means exactly 1/3/4 turns, because the remaining portion just comes out as linker DNA. The DNA lies outside the octamer surface. In these 2 turns, there are about 146 base pairs, and the DNA double helix enters and leaves the nucleosome from the same side of the histone octamer. Okay, so if it is entering from one side, imagine it will complete one round from to the point where it has entered. Again one more round, it will come to the same end where it had entered, and it will leave. So before it leaves, it creates about 1/3/4 turns and comes out as linker DNA. The entire structure is flattened into a disc-like structure, and the histone 1 H1 protein seals off, sticks the two turns of DNA to the octamer or to the nucleosome. One nucleosome is connected to another by short segment of the DNA, I told you, which is called as the linker DNA, having about 50 to 60 base pairs. The entire nucleosome is 11 nanometer thick fiber. The fiber coils around itself, forming a 30 nanometer thick fiber. This is called as the solenoid structure and it forms the chromatic thread. The H1 protein molecules face the inner part of the spiral and interact with each other, giving stability to the solenoid structure. What do you mean? When you had two arms of the chromosomes, the H1 protein is towards the inside of those two arms, thereby giving stability to the entire chromosome which had two chromatids. In one turn of the solenoid, there are about 6 chromosomes. So is this clear?